All right, let's dive deep today. We're going to unlock these things called RTKs.
Oh, yeah. RTKs.
They're like these tiny little switches in our cells, right? And they like control growth.
hmm
And you know what? They're kind of a big deal, especially in cancer research.
Absolutely.
So
Yeah.
we're diving into RTKs. We've got some really cool research papers, even a YouTube video to help us break it all down.
Awesome.
So what are RTKs, really? Like, in simple terms, what's the deal with these things?
Okay, well, RTKs stand for Receptor Tyrosine Kinases,
Okay,
and they're like the communication hubs of our cells. They're not just sending signals, they're like running the show. Like growth, cell division, even like cell death.
whoa.
Yeah, they decide if a cell lives or dies. Pretty dramatic stuff.
So they're calling the shots, huh? Wow, that's right. But how do they actually work? It's not like they're literally flipping switches, are they?
Not quite physical switches, but imagine a lock and key. The key's this thing called a ligand. And when it binds to the RTK, it sets off this process called dimerization.
Dimerization. Okay, break that down for me.
So picture two RTKs, right?
Yeah.
They kind of buddy up like a cellular dance, and they form this pair, a dimer, and that's like turning the RTK on. It can start doing its thing.
Got it. So it's like a power couple, these two RTKs.
You could say that.
Okay, so they're on, ready to work.
Right.
What happens next? I keep seeing this word, autophosphorylation. And it
Ah.
sounds kind of complicated.
It's a mouthful, but it makes sense. Think of it like this. The RTK buddies, they power each other up by adding these things called phosphate groups to themselves, like self-charging batteries.
Whoa.
That's autophosphorylation, self-powered.
So they're not just a switch, they're like self-powering switches.
Yeah.
That's wild.
That's just the beginning. This autophosphorylation thing, it makes special spots in the RTKs, kind of like landing pads.
Landing pads. Okay, what's landing there?
Other molecules,
Got
these
it.
proteins called docking proteins, they're crucial for the next steps.
Docking proteins, more new words. So like what do they do? Like little spaceships landing?
Uh-huh, kind of. They each bind to a specific spot, that landing pad on the RTK dimer.
And these docking proteins, they like tell the cell what to do, right?
Right. Each one sets off a chain reaction, like a signal cascade, telling the cell, grow, divide, even die.
Wow, so this is all getting pretty intense. And it makes me think about the cancer connection we mentioned before. If these RTKs control growth, it makes sense they'd be involved when things go haywire.
Exactly. When the signaling goes out of whack, you get uncontrolled growth, a hallmark of cancer.
So like a broken switch leading to a runaway train of growth. Yikes.
Pretty much. But the good news is, if we understand these RTKs, we can maybe fix those broken switches.
That's where targeted therapies come in, right? I've heard about those.
Right. Scientists are developing drugs to target those malfunctioning RTKs specifically, like cutting off the bad signals.
Throwing a wrench in the machinery, huh?
Exactly. And all these steps we talked about, dimerization, autophosphorylation, those docking proteins, understanding that is crucial for making these therapies work.
It's mind-blowing how much is happening at such a tiny level.
It really is.
But it's not just one kind of RTK, right? There's like a whole bunch of them.
Yeah, it's like a whole family of RTKs, each with their own job and signals. It's a complex network, and we're just starting to map it out.
Incredible. So just to make sure I got it all, these RTKs are like tiny switches. They pair up, power themselves up, and then other proteins land on them to relay instructions to the cell.
You got it. And when they malfunction, things can go wrong. That's where those targeted therapies come in, trying to fix those broken switches.
This is fascinating. I feel like we've only just scratched the surface of the cellular world back again. It's just wild to think about all this complex signaling happening inside our cells, like all the time. Last time, we were talking about those RTKs, those tiny switches, controlling big decisions for the cells. But it feels like we're just getting started. What else is there to uncover about how these things work?
Okay, so let's dive into this idea of microdomains. It's kind of new, even a little bit, controversial, but it's gaining traction for sure.
Microdomains. So like little neighborhoods inside the cell, like gated communities where certain molecules hang out.
Not a bad analogy. The idea is that these signaling pathways, especially ones with RTKs, they're not just like randomly scattered everywhere. They're organized into these microdomains.
So there's actually some structure to this whole molecular world. It's not just chaos in there.
Exactly. It's all about efficiency, specificity, kind of like departments in a company, each with their own specialty.
Makes sense. If everything was just bumping around randomly, things could get messy. But how do these microdomains form? What keeps everything in its place?
Well, there are different mechanisms, but one key player is this protein called, get this, kinase anchoring protein, or AKP.
Sounds like something you'd find on a playground.
I know, right? Funny name. But don't be fooled. These AKKPs are super important. They're like scaffold proteins, keeping everything organized in those microdomains, like the architects, making sure the neighborhood's built right.
So they're bringing all the players together. But why is this organization so important? What difference does it make if things are clustered or spread out?
Imagine trying to talk in a room where everyone's shouting. Impossible to hear, right? But smaller groups, focused conversations, much better.
Yeah, so it's like having a dedicated meeting spot instead of everyone yelling across a crowded room.
Exactly. Those microdomains create a more concentrated signal, makes things more efficient. And that specificity is crucial, especially for things like cell growth and division. You don't want mixed messages there.
Wow, so much organization at such a tiny scale. It's mind-blowing how complex cells are.
It really is. And the more we learn about these microdomains, the more we see how they could be manipulated. Huge implications for treating diseases, especially cancer.
Right, because in the last part we talked about how messed up RTKs can lead to cancer.
Yeah.
Could these messed up microdomains also play a role?
Good thinking. You're spot on. Disruptions in those microdomains can totally throw off the balance of signaling, and that can mean uncontrolled growth, potentially leading to cancer.
So it's not just about faulty switches, but also a messed up communication network.
Exactly. It's like having a city with broken traffic lights and roads leading nowhere.
And I'm guessing some researchers think we could actually target these microdomains as a cancer treatment strategy.
Right. It's like fixing the city's infrastructure, not just the individual cars. Still early stages, but very promising.
So this knowledge about RTKs and their organization, is it already leading to better treatments?
Definitely. More targeted therapies. Hitting those malfunctioning RTKs, disrupting those bad pathways, and the results are pretty amazing.
So we're using this knowledge about these tiny switches to develop actual life-saving treatments. That's incredible.
It really shows how important basic research is. Understanding these fundamental things can lead to huge breakthroughs.
And this is just the beginning, right?
Oh yeah, tons of research still happening. Scientists are working hard to decode these systems, figure out their full potential.
What are some of the big questions they're tackling right now?
One big one is, what are the specific roles of each RTK family in different cancers? Remember, there are over 50 of these things, each with their own quirks. It's like learning the language of each individual switch.
So it's not one size fits all. Each type of cancer might need a different approach depending on which RTK is involved.
Precisely. Figure out which RTK is driving the cancer, and you can tailor the treatment. More effective, fewer side effects. That's personalized medicine in action.
And I bet understanding those microdomains is another big area of focus, right?
Absolutely. The more we know about how they regulate RTKs, the more options we have for treatments, like discovering a whole new toolkit.
Wow, it sounds like a revolution in cancer therapy is just around the corner. It's amazing how far we've come in understanding these cellular processes. And we're back. For the final part of our deep dive, we've been exploring these RTKs, those tiny little switches that are so important for cells, especially in cancer.
Yeah. Dimerization, autophosphorylation, docking proteins, even those microdomains. It's complex, but it all works together so beautifully.
Absolutely. But now we're switching gears a bit. We're going to zoom in on a different type of cellular communication, the role of glycine receptors. I'll be honest, I don't know much about those. What are they and how are they different from these RTKs we've been talking about?
So glycine receptors, they're a type of neurotransmitter receptor mainly found in our central nervous system. Think of them like specialized receivers, picking up signals between nerve cells, neurons.
So they're like part of the communication network in our brains and spinal cord. That sounds pretty important.
Oh, it is. They help transmit signals across synapses, those tiny gaps between neurons, like a relay race, passing the baton smoothly between runners.
Okay, I'm getting a picture. But what about glycine itself? What is that exactly?
Well, glycine is actually an amino acid, one of the building blocks of proteins.
Okay.
But it also acts as a neurotransmitter, a chemical messenger carrying signals between those nerve cells.
So glycine is the message. The glycine receptor is the receiver, kind of like a lock and key.
You got it. And when glycine binds to its receptor, it changes the receptor's shape. This allows ions, these tiny charged particles, to flow across the cell membrane.
Hold on, ions. What do those have to do with it?
Think of them like tiny electrical signals. Their movement changes the electrical potential of the neuron, which can either excite or inhibit its activity.
Wow, so it's not just a message, it's controlling electricity in the nervous system. That's super precise.
It is. And in the spinal cord, glycine mainly acts as an inhibitory neurotransmitter.
Inhibitory, so like a brake pedal, slowing things down.
Exactly. It helps regulate muscle movement, prevents those motor neurons from firing too easily.
Okay, so we talked about uncontrolled growth in cancer. Could these glycine receptors also be involved in diseases, maybe those affecting the nervous system?
Great question. You're totally right. When glycine receptor signaling goes wrong, it can contribute to neurological disorders. Like a faulty brake system, things can get out of control.
Makes sense. So what kind of disorders are we talking about?
One example is hyperexplexia, sometimes called startle disease.
Oh, I've heard of that.
People have these exaggerated startle responses, even to things that wouldn't normally cause a big reaction.
So like someone might jump or flinch at small sounds or touches.
Exactly. And research shows that mutations in the genes for glycine receptors can mess up their function, leading to that heightened startle reflex. It's like the off switch for the startle response is broken.
Wow, so these receptors are really important for our nervous system to work properly. It's amazing how a tiny problem at the molecular level can have such a big impact.
It really shows how interconnected everything is, and how important it is to keep researching these basic mechanisms. The more we understand, the better we can treat these disorders.
Are scientists working on new treatments that specifically target these receptors?
Yeah. They're looking at different strategies, like drugs to enhance glycine receptor activity. Boosting that brake pedal effect could be really helpful for conditions like hyperexplexia, where that inhibitory signaling is messed up.
It's been amazing, this journey into the molecular world. We started with those fascinating RTKs, their connection to cancer, even those microdomains. And now we've explored glycine receptors, how they control communication in the nervous system, and how problems with them can lead to neurological disorders.
It's incredible, isn't it? All this happening inside us right now. All these signals controlling our thoughts, movements, everything.
It's a reminder that even though we can't see it, there's this whole universe of activity at the microscopic level. And by understanding it, we can find new ways to treat diseases and improve people's lives.
Absolutely. It shows the power of scientific curiosity and the potential that's inside each and every cell.
Well, that wraps up our deep dive into the world of cellular signaling. We've covered a lot, but as we've seen, there's always more to learn. So until next time, stay curious about the world around you. And remember, even the smallest things can have a huge impact.